Your last meal was hours ago. The kitchen is closed, the glucose is running low, and something shifts deep inside your cells. Quietly, without any signal you'd consciously notice. This isn't starvation. This is your body switching operating systems. Within the first 12 to 16 hours of fasting, glycogen stores in the liver begin to deplete. Insulin drops, and as insulin falls, a cascade of cellular events begins that most people never learned about in any health class. The body starts tagging damaged proteins, broken organelles, and cellular waste for recycling, a process that has been running in human biology for millions of years, long before anyone gave it a name. That name, of course, is autophagy. And while most conversations about it focus on fat loss or longevity, the place where its effects are perhaps most misunderstood and most fascinating is inside your muscle tissue. Because what happens to your muscles when autophagy peaks isn't what most people expect. And by the time this video is over, you'll understand exactly why. Picture a muscle cell as a factory that never fully shuts down. It's constantly producing, contracting, repairing, and generating waste in the process. Damaged proteins accumulate. Mitochondria wear out. Cellular debris builds up in ways that quietly compromise function over time. Autophagy is the factory's cleaning crew, and fasting is what calls them in to work the night shift. Inside skeletal muscle, autophagy operates through a highly regulated system involving proteins like BCLIN1, LC3, and the ULK1 complex, molecular machinery that identifies damaged components, wraps them in a double membrane structure called an autophagosome, and delivers them to the lysosome for breakdown and reuse. Research by Masaki Komatsu and colleagues demonstrated that when autophagy is genetically blocked in mouse muscle tissue, the animals develop progressive muscle weakness and accumulation of abnormal protein aggregates, even without fasting. In other words, autophagy isn't optional maintenance. It's how muscle tissue survives itself. What makes this particularly striking is that muscle cells are among the most autophagy-active tissues in the entire body during fasting. A 2013 review by Sandry and colleagues in Nature Reviews Molecular Cell Biology outlined how skeletal muscle relies on autophagic flux, not just for cleaning, but for energy homeostasis, breaking down internal components to fuel contraction when external fuel isn't available. The muscle isn't being cannibalized, it's being managed. Here's the part that surprises most people. Fasting, even extended fasting, does not automatically cause significant muscle loss, and the science behind why that's true is more interesting than the fear itself. The assumption comes from a logical sounding chain. No food means no protein intake. No protein means muscle breakdown. Muscle breakdown means you're losing the tissue you worked hard to build. But that chain skips several steps. The body has a sophisticated prioritization system. During early fasting, the primary fuel sources are liver glycogen and fatty acids released from adipose tissue. Muscle protein catabolism, actual muscle breakdown for energy, is a metabolic last resort, not a first response. A landmark study by Cahill and colleagues, originally published in the 1970s and repeatedly referenced in modern fasting research, mapped the sequential fuel hierarchy during starvation and confirmed that significant muscle protein is not mobilized until fat stores become critically low. What autophagy adds to this picture is a layer of internal recycling that actually reduces the need to break down structural muscle protein. By dismantling damaged intracellular components and recycling their amino acids for immediate use, autophagy gives the muscle cell a way to meet part of its protein needs without touching the contractile fibers themselves. The muscle isn't losing ground. In many cases, it's quietly reorganizing. The real question is what happens when you push further, and when exactly autophagy in muscle tissue actually reaches its peak. That's exactly where things get interesting, and we'll get there. One of the most common questions in fasting research is deceptively simple. When does autophagy peak? The honest answer is that it depends on the tissue, and muscle follows its own schedule, distinct from liver or brain. Understanding that timeline changes how you think about fasting windows entirely. In skeletal muscle, autophagy markers begin rising measurably around the 16 to 18-hour mark of fasting, based on studies measuring LC34 flux and C62 degradation in human and animal models. Research published in Autophagy examining autophagic activity in human skeletal muscle demonstrated that these markers increased significantly after just a single overnight fast, with activity continuing to climb through the 24-hour window. Peak autophagic flux in muscle tissue, meaning the highest rate of autophagosome formation and lysosomal degradation, appears to occur somewhere between 24 and 48 hours of fasting in most human studies, though individual variation based on metabolic rate, prior nutrition, and training status affects that window considerably. What's critical to understand is that peak doesn't mean the process suddenly stops being useful after that point. It means the rate of autophagic activity reaches its highest measurable intensity before the body begins compensatory adaptations. Beyond 48 hours, the body starts modulating the process, dialing certain pathways up or down based on cellular stress signals, which is part of why extended fasting research produces such complex and sometimes contradictory data. The muscle, in other words, doesn't just sit passively through this. It responds. Of everything autophagy does inside a muscle cell, perhaps the most consequential process is one most people have never heard of: mitophagy. It's a specific branch of autophagy targeted exclusively at mitochondria. And in muscle tissue, it may be the most important cellular housekeeping event that fasting triggers. Mitochondria in muscle cells are under constant oxidative stress. Every contraction generates reactive oxygen species. Over time, damaged mitochondria accumulate, and dysfunctional mitochondria don't just underperform. They actively leak electrons, generate excess oxidative damage, and can trigger cell death pathways. Mitophagy, regulated in large part by the Pinkin-Parkin pathway, selectively identifies and eliminates these compromised mitochondria before they cause broader cellular damage. Research on fasting-induced mitophagy in skeletal muscle has consistently demonstrated that this process is not a sign of deterioration. It functions as a quality control mechanism that clears the field for healthier mitochondrial populations to expand through a complementary process called mitochondrial biogenesis, a finding that has emerged consistently across the literature examining mitochondrial quality control in skeletal muscle. The implications for muscle function are significant. A muscle operating with a higher proportion of healthy, efficient mitochondria contracts more powerfully, fatigues more slowly, and recovers faster. This is part of why the relationship between fasting and athletic performance has become an area of active scientific inquiry, with researchers examining mitochondrial quality control as one of the mechanisms through which fasting may influence muscle physiology over time, though the literature in this area remains early stage. What's already clear from the existing literature is that mitophagy during fasting doesn't weaken muscle at the cellular level. It selectively removes what was already compromising it. Not all muscle fibers respond to fasting the same way, and this distinction rarely makes it into mainstream conversations about autophagy, even though it fundamentally shapes what's actually happening in your body during an extended fast. Human skeletal muscle contains two primary fiber types: type 1, slow twitch fibers optimized for endurance and sustained low-intensity work, and type 2, fast twitch fibers designed for power, speed, and high force output. These two populations have different metabolic profiles, different mitochondrial densities, and critically, different autophagy sensitivities. Morphological research characterizing fiber-type metabolic profiles has established that type 1 fibers, with their higher mitochondrial content and reliance on oxidative metabolism, show earlier and more robust autophagic responses during fasting, a pattern consistent with autophagic flux measurements documented in human skeletal muscle research by Jamart, Mashalain, and colleagues examining fiber-specific responses to metabolic stress. Type 2 fibers, being more glycolytic, respond somewhat differently and appear more vulnerable to atrophy signaling through the mTOR-FOXO3 axis during prolonged caloric restriction. This fiber type specificity matters because it means fasting doesn't act uniformly on muscle. The slow-twitch fibers used in daily movement and postural stability tend to handle autophagic stress well. The fast-twitch fibers responsible for explosive strength are the ones that require more careful consideration in the context of very extended fasting, particularly beyond 48 hours, where protein synthesis rates and anabolic signaling begin to shift in ways that could compromise fiber volume over time. This fiber type distinction is one the research documents clearly. Fasting at different durations places different demands on different parts of the muscle system, and the literature on fast-twitch fiber response beyond 48 hours reflects that complexity. And the hormonal story behind all of this makes it even more layered. If you want to understand what really protects muscle during a fast, you have to follow the hormones. Because while autophagy is doing its cellular work, there's a hormonal negotiation happening in parallel. And the outcome of that negotiation determines how much muscle you actually preserve. Growth hormone is the protagonist of this story. During fasting, growth hormone secretion increases dramatically, sometimes by 3 to 5 times baseline levels, according to research by Ho and colleagues published in the New England Journal of Medicine as far back as 1988, findings that have been repeatedly validated in more recent literature. Growth hormone doesn't directly build muscle during a fast. There's not enough circulating IGF-1 to drive significant anabolism in a calorie deficit state. But it does something arguably more important. It preserves lean mass by shifting the body toward fat oxidation and away from muscle protein catabolism. It's a lipolytic signal that protects structural tissue by making adipose tissue the preferred fuel source. Cortisol, however, is the counterforce. As fasting extends past the 24-hour mark, cortisol levels rise as part of the body's broader stress response. And cortisol is catabolic, meaning it can accelerate protein breakdown, including in muscle tissue. The balance between growth hormone's protective signaling and cortisol's catabolic pressure is one of the key determinants of whether extended fasting preserves or begins to erode muscle mass at a meaningful rate. This is precisely why the research doesn't give a single clean answer about fasting and muscle. It's looking at a dynamic, shifting hormonal environment where timing, individual physiology, and fasting duration all interact. And what happens when researchers actually try to measure the outcome of all this? That's what the data shows. And it's more nuanced than either side of the fasting debate usually admits. The scientific literature on fasting and muscle mass is more nuanced than most fitness content suggests. And reading it carefully reveals a picture that neither extreme of the debate accurately represents. The fear that fasting inevitably destroys muscle is not well supported by the evidence. Neither is the claim that fasting has zero effect on lean mass under any circumstances. A 2011 systematic review by Putani and colleagues examining alternate day fasting protocols found that participants retained lean mass remarkably well over periods of 3 to 12 weeks, with fat mass accounting for the overwhelming majority of weight loss. A more granular analysis published in Nutrition Reviews by Tinsley and LaBounty in 2015 specifically examined intermittent fasting's effects on lean tissue and concluded that short to moderate duration fasting, up to 48 hours, does not appear to cause clinically significant muscle loss in healthy adults with adequate baseline protein status. The body's conservation mechanisms, including the growth hormone response and autophagy-driven internal recycling discussed earlier, appear sufficient to protect structural muscle protein during these windows. Where the research becomes more cautious is in the context of very extended fasting, beyond 72 hours, and in individuals who combine prolonged fasting with high training volumes without adequate refeeding protocols. Research measuring muscle protein synthesis rates during extended fasting has documented a measurable decline after 48 hours, even in cases where lean mass measurements remain stable in the short term, indicating that suppression of synthesis and net tissue loss are distinct phenomena that the literature treats separately. This distinction between acute suppression of synthesis and actual net muscle loss is one that the literature handles carefully, and one that popular content almost never addresses with the precision it deserves. One of the most important conceptual distinctions in this entire conversation is one that gets almost no attention: the difference between muscle degradation and muscle renovation. These two processes can look identical on the surface. Both involve the breakdown of muscle protein, but they represent fundamentally opposite outcomes for the tissue. Muscle degradation, in the pathological sense, refers to net loss of contractile protein, the actin and myosin filaments that give muscle its functional structure. This is what occurs in conditions like cachexia, prolonged immobilization, or severe caloric restriction over weeks and months. Muscle renovation, by contrast, refers to the selective removal of damaged, misfolded, or dysfunctional proteins within the muscle cell, replacing compromised material without net loss of functional tissue. Autophagy, at its core, is a renovation process. Research by Sandry published in Physiological Reviews in 2013 drew this distinction explicitly, noting that basal autophagy in muscle is essential for maintaining fiber integrity, and that its suppression, not its activation, leads to the accumulation of damaged proteins associated with muscle weakness and disease. This reframing matters because it changes how fasting-induced autophagy in muscle should be interpreted. When autophagic flux increases during a fast, the muscle is not being dismantled. It is being selectively edited. The structural proteins that define muscle function, actin, myosin, the contractile apparatus, are not the primary targets of this process. What autophagy addresses is the accumulated cellular noise, misfolded proteins, dysfunctional organelles, oxidatively damaged material that compromises the fiber's operational integrity. The muscle that exists on the other side of peak autophagy is not the same muscle that entered the fast. And understanding what it actually looks like at that point is where this conversation ends up. If you've made it this far and found the science genuinely useful, drop a comment below. What surprised you most about how autophagy actually works in muscle tissue? The answers people leave on videos like this one are always worth reading. The end of a fast isn't just the return of food. In terms of muscle biology, it's the beginning of a remodeling window, a period where the cellular environment inside the muscle has been reorganized by the autophagic process and is, in specific measurable ways, different from what it was before the fast began. Research examining post-fasting muscle physiology has documented several consistent findings. Mitochondrial quality improves following fasting-induced mitophagy, with studies showing increases in markers of mitochondrial efficiency and reductions in oxidative damage in skeletal muscle after fasting periods sufficient to activate robust autophagic flux. Protein aggregate clearance, the removal of misfolded and damaged proteins that accumulate with age and exercise, is measurably improved. And critically, the anabolic sensitivity of muscle tissue appears to increase following a period of fasting. meaning that the muscle's response to protein and resistance stimulus in the refeeding window is enhanced compared to a continuously fed state. A study by Deldick and colleagues published in the European Journal of Applied Physiology documented increased activation of mTOR signaling in skeletal muscle following refeeding after fasting, suggesting the muscle is primed to rebuild efficiently once nutrients return. What this body of research collectively describes is not a muscle weakened by the fasting process, but a muscle that has undergone selective internal maintenance, cleared of accumulated damage, operating with a cleaner mitochondrial pool, and positioned to respond more efficiently to the anabolic signals that follow refeeding. That outcome is not guaranteed under all fasting conditions or for all individuals. And the research is clear that duration, protein status, training history, and refeeding quality all shape the final result. But the picture that emerges from the science is one of cellular renovation, not deterioration. And that distinction is worth understanding precisely because the popular conversation around fasting and muscle almost never gets this specific. If this video gave you a clearer picture of what's actually happening in your muscle tissue during a fast, share it with someone who still believes fasting automatically breaks down muscle, because the science tells a very different story. Liking the video and subscribing helps this kind of research-based content reach more people who are looking for answers that go beyond the surface.